In Chapter 5.5, we're going to talk about a few special structures that bacteria can make. We'll talk about pili and stalks, which allow bacteria to adhere or attach to a surface, particularly when the environment is favorable to them. Remember, this is one of the things that pathogens have to do. When they get inside of the host, they have to attach to a surface, otherwise they get excreted. We'll also talk about flagellar motion. So flagellae in bacteria actually turn. They have a little motor, basically, that turns them, and they can propel the bacteria through a liquid. And we'll talk about a related process called chemotaxis, which allows bacteria to respond to and basically sense things in their environment and move to or away from them. Okay, bacterial cells respond to their environment. That shouldn't be a surprise, right? Every organism responds to its environment. It's going to move away from things that are harmful to it, and it's going to move towards things that are good for it. In the case of bacteria, they generally often want to adhere or attach to a specific surface. So adherence is the ability to attach to a substrate. That means a surface. Say you're a bacteria in a nutrient-rich pond and you want to attach to a rock that's just right in the perfect zone of getting just enough sun and just enough nutrients, right? You need structures to do that then. Cells in a nutrient-rich host, say you want to move through your host and go to a place like, I don't know, the intestines, where there's lots of nutrients. You have to be able to move there and then adhere to that location to stay there. Sometimes, though, the environment might change, so you have to move. This is called motility, the ability to move and relocate. So you have to be both motile in some cases and be able to adhere in others. Some bacteria are constantly motile, some are very rarely motile, some adhere quite frequently. Just depends on the species and the conditions. So we're going to talk about structures called pili and stalks. So pili is technically plural. A pilus is the singular. These are made of a protein called pilin. That's why they're called pili. And they're basically large kind of, projections that come off of the cell. There's some called fimbriae, which are short attachment pili. They help attach to surfaces. There's a special pilus called the conjugation pilus. Sometimes it's called the sex pilus, but that's an improper term for it because bacteria don't have sex. They asexually reproduce, but we'll see this later. This conjugation pilus can help transfer DNA between one bacteria and another. Now, why do we care about that? Well, if one bacteria has a gene that allows it to resist antibiotics, through this process, it can transfer that to another bacteria. That's called horizontal gene transfer. That's like you giving some of your DNA to your cat, or vice versa. Your cat giving you some of its DNA, and now you could have cat ears or something like that. I don't know. It's something weird to think about, but it happens in the bacterial world all the time. We also have a set of things called stalks. These are kind of extensions of the membrane and the cytoplasm that secrete adhesion factors, proteins that help the bacteria stick. So these are actually projections, and this is like the membrane budding out from the cell. So here's an example. Here are some pili, and then these would be the fimbriae, the small ones. And then we'll talk about this structure next, a flagella. In bacteria have evolved what we call a rotary flagella, which spins kind of in a corkscrew manner, acting a bit like a propeller to enable motility, and this process called chemotaxis, which I'll define in the next slide. Some bacteria have multiple flagellae, some only have one. These are different from eukaryotic flagella. You might think of a sperm cell, right? That has a flagellae. It has a flagella, singular. But the flagella in sperm evolved completely independently from the bacterial ones. So the bacterial ones twist, whereas eukaryotic ones kind of undulate up and down. So they evolved completely differently. That's called convergent evolution, when kind of you solve the same problem 2 different times. The thing that's really cool about flagella is they're like a little motor. Like, this is a biomolecule or biomolecules, but it's shaped like a motor. I think this is the coolest thing. Here's an electron micrograph of it. It looks like an attachment that we would have for some sort of like propeller or something like that. And it actually spins around and helps the bacteria drive through the water. So motility, it's not just random. You might not think of bacteria as being smart, and they're not really, right? They don't have a brain, they don't really have a nervous system, but they can sense their environment. And this is called chemotaxis. So chemo means chemical, and taxis means movement. This is sensing chemical signals to move towards or away from them. Chemotaxis is super cool. There's all kinds of different things. There's different types of taxis. There's phototaxis, moving towards light. There's magnetotaxis. I'll show you a link to a video you can watch where some bacteria can sense magnetic fields and they'll move towards or away from them. So some scientists set up a little magnet and they hooked it up to a Nintendo controller and they can control the direction the bacteria will swim by moving the magnet around. Super cool. This chemotaxis is really critical to sensing the environment and moving towards food and away from toxic molecules. And it coordinates with the flagella. So bacteria actually have receptors on them. You can think of these kind of like taste or scent, right? You can smell things because you have receptors in your nose, and you can taste things with the ones in your tongue and in your nose. Taste is slightly scent as well. Bacteria can taste the chemicals in their environment with these receptors, and then they can coordinate their movements. So they have a flagella, right, that rotate as propellers, and they do a semi-random movement. It's only semi-random, it's not fully random. So they'll move towards something, and then they'll sense, and then they'll move in a different way and kind of go around. If they're not sensing that they're getting closer, they turn. Okay, we're not, well, starting to, let's try a different way. Okay, not really getting closer. Oh, we're definitely not. And then they'll move towards it eventually. So they kind of move in a semi-random way, but eventually they will move towards something that attracts them and away from something that repels them. So we would call this, in the case of moving towards something, positive chemotaxis, moving towards what we could call a chemoattractant. And negative chemotaxis is moving away from things that are chemorepellents. This could be, I don't know, the immune system or things like that. And this definitely would be like food molecules. So I can't show the video because it's on YouTube and I post the video on YouTube, but the link is here. It's super cool. It's a funny video too. They're a great group of scientists that actually hook the magnet up to a controller and they move the bacteria around with the magnet. So go check that out when you got some time. Okay, we've talked about this last piece a little bit before, but I want to talk about a structure that some bacteria can make called an endospore. This is a protective coating. It protects bacteria from heat and chemicals. It's like a resistant shell. And when the bacteria form this, they kind of go dormant, and these spores can survive for a long time. In fact, they can survive so long that some scientists found bacterial spores buried in amber. Yes, Jurassic Park, literally. They found spores in there. They were able to revive them and grow million-year-old bacterial spores that had been preserved in amber. So that's pretty sweet, right? No dinosaurs, but ancient bacteria. So these bacterial endospores get made when the bacteria senses that things are not going its way, right? The environment is not favorable. We need a protective shell that can resist heat and chemicals. Now, medically, these are very dangerous because you can't kill these with standard cleaning agents. You could apply bleach to them and it would resist it. The only way to get rid of these really is to autoclave something. So we have several species that we've heard of. Bacillus, right? Bacillus anthracis, which causes anthrax. It makes spores. And Clostridium, we saw Clostridium botulinum, which made the botulism toxin. It makes spores too. So they're very difficult to get rid of. And that means they're difficult to stain. To stain these, you have to use a stain called malachite green, and you actually heat them up for quite a while. It penetrates into the spore, and you can decolorize it and then counterstain what we call the vegetative part. So this spore grows off of another part of the bacterial cell here. That's just a small smattering of the different structures that bacteria can make. They have a whole other specialized group like thylakoids and things like that, but we're not going to get into those. The main thing is that bacteria need to make structures to adhere, pili and fimbriae. These are protein filaments, or they can make stalks, which are extensions of the cell membrane. This enables them to stay in a favorable environment. If they need to move, flagellar motility, moving with those rotary flagella, is very useful. And they're going to participate then in chemotaxis, kind of random, semi-random walking up a concentration gradient towards an attractant or down a concentration gradient away from a repellent. And then we have endospores, our heat and chemical resistant dormant stage structures.